Okay, so hi everyone. Welcome to the physical property of the cytoplasm seminar series, and this is our first session semester of 2026, and it's also our 43rd session overall. And It's a pleasure to introduce Professor Sean Sun from Johns Hopkins University. Sean studied at Penn State and received his PhD in theory from UC Berkeley. And he is now a professor of mechanical and biomedical engineering, studying the physics of cell movement, cell shape, and cell size, as well as fluid transport across the cell membrane. Today, he'll speak about how membrane voltage gradients are established in migrating cells. Sean, thank you for joining us, and the screen is yours.
Thank you so much, Yuping. Thank you for giving us the opportunity to tell you about our work. This work is, somewhat fresh. We're still working on some aspects of this, but some of the results are pretty interesting, I think, and informs on the physical properties of cytoplasm, I think. Um, so uh, Before I go on, I really should acknowledge the people that really are responsible for this work. This work is really the outstanding, the result of uh, the work of outstanding graduate student, Zhuozhu Ge. He's a graduate student in my lab, and also a postdoc in my lab, Qing Ni. Uh, you know, Qing and Zhuozhu both did the experimental part of this. A lot of the experiments you see in this paper. And then some of the modeling work is done by Yufei. Yufei was a PhD student in my lab. He's now graduated. Um, so they together collaboratively made this work possible. Qing will be on the job market. He's a postdoc, so he's he's going to be on the job market in 2027. So he's going to be looking for a faculty position. And I also should acknowledge people from the Constantopoulos lab and Ding Chang Ling's lab. They helped us with some of the experimental methods that I'll present in this work. Okay. All right. So my lab, we actually been kind of interested in cell ionic homeostasis for a long time. Uh, the idea that the cells try to maintain some sort of ionic environment for all the important cytoplasmic processes. This question, personally, I think, is really at the heart of a lot of different questions in cell biology. We came to this realization initially when we first tried to, look at how cells, regulate their volume and how cells respond to mechanical forces. I mean, I came from the mechanobiology biology field. We kind of are interested in the question of how cells respond to mechanical forces. Very soon we realized that this question is tied together with cell ionic homeostasis. And that's because, ions are the most abundant species in the cytoplasm, and different kinds of ions, especially things like potassium, sodium, are in high concentrations. And together with proteins and amino acids, they ultimately determine some of the important physical parameters in the cytoplasm. So the parameters are the osmotic pressure, pi, which come from, solute mixing with water, and also the hydraulic pressure, P, which is different from osmotic pressure, uh, which is just the hydrostatic pressure in the cytoplasm. So these variables will determine the overall cell water content, which of course will then determine the concentration of proteins. And also, because hydrostatic pressure is connected with externally applied force, right? So if you apply force to the cell, for example, it will influence hydrostatic pressure in the cytoplasm. This is also central to mechanobiology. So the question, mechanobiology or cells, how cells respond to mechanical forces, cannot be decoupled from the question of cell ionic homeostasis. And then because, uh, ionic concentrations ultimately determine protein concentrations, this question is also related to cell mass density, which has been uh, kind of a recent interest in the field, right? There's been a lot of interesting papers on cell mass density and how cell mass density potentially influences questions like uh, cancer and aging. Uh, so cell mass density is related to ionic homeostasis because if you change ionic concentrations, you will impact protein concentration. And in some papers that we published recently, we showed that this cell mass density, CMD, and also nuclear mass density will also impact protein translation and growth. And then finally, this is probably something that everyone kind of knows intuitively, but very difficult to fully disentangle, is that global gene regulation is also related to cell ionic homeostasis, simply because the presence of potassium will influence chromosome compaction, right? DNA is highly charged, and if the counterions are not interacting with, or counterions like potassium interacting with DNA, and that will influence the conformation of DNA. And we now know that conformation of chromosomes determine gene regulation and lots of other questions of the cell. So hopefully you share my enthusiasm for this topic of ionic homeostasis because it really is one of the most important questions in the cell. So in the question of cell ionic homeostasis, membrane voltage plays very prominently because membrane voltage will, the transmembrane voltage will influence ionic flux across the membrane, right? Because ions are charged, if you have a voltage difference across the membrane, it will influence ionic charge and therefore impact ion concentrations in the cell. So this is kind of a chicken and egg question because ion pumps like sodium potassium exchanger sets up the sodium potassium gradients across the cell membrane. You probably know that there's high concentration of potassium in the cytoplasm, but low concentration of sodium, and this is set up by the action of sodium potassium exchanger. And this ion concentration then and the resulting fluxes that's across the membrane sets up a membrane potential difference about minus 70 millivolts across the membrane for non-excitable cells, right? So for excitable cells like neurons, this can spike into a much higher value, but for non-excitable cells, it's on the order of minus 70 millivolts. So the cell cytoplasm is slightly negative, and it's the presence of this membrane potential is really because I have various different types of ion channels passaging sodium, potassium, and other ions across the membrane, and that flux depends on the presence of membrane voltage, and these two factors will work together to set up this homeostatic membrane voltage at the resting state. So just to give you a little scale of reference, the thermal membrane voltage, in other words, the amount of voltage that's contained in thermal energy, is on the order of 26 millivolts. So that's just coming from RT divided by the Faraday constant. This you see is just so the resting membrane voltage for non-excitable cells is just a few kBt in energy scale. It's not very large, but this translates to a very large electric field because this is occurring over presumably 5 nanometers of distance. And the membrane voltage is obviously important for ionic fluxes, and by the same token, it influences all charged solute flux. And this includes amino acids because amino acids are typically charged, and if they're transported together with counterions, then their transport flux also depends on the ionic concentration of counterions. So the question of membrane voltage ultimately also influences things like metabolism. So there is a biophysical model of membrane voltage, so-called the Goldman-Hodgkin-Katz model of membrane potential. And this just basically follows a couple of different assumptions. One is that cell arrest must have net zero flux, right? So total flux of potassium is zero, total flux of sodium is equal to zero at rest. And also the cytoplasm must be electron neutral. In other words, I don't have uh, positive or negative charges in the cytoplasm. You may have excess charges on the membrane. So all the excess charges actually coming from uh, positive potassium ions are sitting on the membrane, and that's what sets up the membrane potential. So in the end, if you follow these two assumptions, you'll arrive at some sort of expression for membrane potential that depends on the thermal membrane potential scale and also the concentration gradient across the membrane, sodium potassium gradient inside outside, other ions, and also permeability of various different types of ions through the membrane. So in this expression, you see that everything depends on ion concentrations and also ion channel activity, which allow the ions go across the membrane. So following this biophysical model, then the prediction will be that because ions diffuse really rapidly, milliseconds, ions are basically uniform in the cytoplasm. This says that the membrane potential should be basically uniform around the cell periphery, right? There is a gradient across the cell membrane from inside outside, but there should not be a gradient around the cell periphery because ion concentrations should be more or less uniform across the cell membrane. Now you can relax one of the assumptions a little bit, right? So I said that one of the assumptions here is that the net flux of ions is equal to zero. There are situations where the net flux is not equal to zero. This is some work we did some years ago, so-called the osmotic engine model. The idea here is that if you have ions coming into the cell at one region and leaving the cell another region, so for example, sodium chloride is coming in at the front, going out the back, then this solute flux can drive water flux across the cell and then drive cell movement. So in this situation, you see that the net flux of sodium and chloride is not equal to zero. There's some small net flux. But from this model, you can actually estimate or calculate how much membrane potential gradient you expect, right? Because I have different, so this is a plot of the polarization, for example, of the chloride channel at the front versus back, leading edge versus back. If you have excess chloride channel, for example, in one region versus another, this will generate non-zero net flux of chloride. And you can ask, how much membrane potential gradient you expect from the front and back with a polarized chloride channel activity. Okay, and then the result is actually quite simple. We expect some gradient, but it's not significant. The gradient is at most a few millivolts, and that's smaller than the 26 millivolt of thermal gradient that you expect, right? So if you relax the assumption that the net flux is equal to zero, you do get a small gradient, but you don't expect to see a large gradient. Certainly not at a very easily observable gradient. So yeah. So there's been actually quite a lot of interest in membrane potential recently, right? So I'll just highlight some papers. This paper coming from Marcus Poussin's lab that showed that in epithelia, membrane potential is related to cell proliferation. This whole paper is worth a read. There are a lot of interesting results there, connecting membrane potential, lots of different cellular processes. Another paper recently, just randomly picked out, showed that if you deplete ATP, there's cell depolarization, and then this ultimately leads to apoptosis, cell death. And then membrane potential has been known to, drive or influence lots of processes of development and cell migration, right? This is now classic work coming from Michael Levine's lab, where you you take a flatworm, you cut it in half, and then you change the ionic environment, or you add in ionophore. Uh, then this leads to altered development of the worm. You even get this double-headed phenotype, which is super interesting, right? And then more recently, my colleague Peter Devriotis in his lab showed very clearly that if you change uh, the charge environment uh, on the cell membrane, right, using a charged peptide that binds to the membrane, you can influence cell migration. Some electric fields been known to influence cell migration for a long time. So clearly, the question of electric field is also related to cell migration mechanisms. Okay, so we decided to take a look at the cell membrane voltage using a fluorescent reporter uh, developed by Francois Saint-Pierre's lab. So they have this reporter called JEDI, which is a membrane protein that has a voltage sensing domain. And then we decided to make this into a ratiometric version using a fluorophore CpGFP, which responds to the voltage domain movement, right? So the fluorescence of GFP will change if the voltage changes. And then there's also a reference, a fluorophore CYOFP, whose fluorescence doesn't change. This allows us to mark the local abundance of the membrane reporter in the cell membrane. Ratiometric probe is important because this removes some of the imaging artifacts due to various different kinds of membrane localization problems or also point spread function issues. So if you then express this ratiometric probe in the cell, immediately you see that there's really quite a lot of spatial heterogeneity in the cell membrane. So this is CYOFP channel. This is the JEDI channel, which is the GFP fluorescence. And then this is the ratio. The ratio is low. You see it's red. Red means low at the protrusive region of the cell. And then the high means green or more yellow at the lateral region of the cell. So the lateral region is on the side of the cell. This is on glass, but also the same thing you see on the when the cell is in 3D collagen. This is 3T3 cells, so presumably naturally they like to be in 3D collagen matrix. You see the same kind of gradient in a collagen matrix. And then you can quantify this ratio, the JEDI ratio around the cell periphery, plotting at different regions. You see that the differences in ratio is quite large, leading and trailing edges versus the lateral regions, okay, show quite a bit of difference. This is not because somehow, JEDI is locally expressed at a high level for some reason, right? Not an artifact of those things, because if you look at the channels separately, you do see a spatial variation in CYOFP, right? Some regions is high in CYOFP level, but the JEDI fluorescence is low, right? And that leads to a different ratio. So the ratiometric probe we think is working, is telling us locally what the membrane potential should be. And then you can do a 3D Z-stack. This is actually coming from a deconvolution of 3D Z-stack using Richardson-Lucy deconvolution. And it also shows exactly this result, which is the leading edge. The protrusive region of the cell is depolarized. The lateral region of the cell is hyperpolarized, and the trailing edge is a little bit also depolarized. So we do this quantification by masking the cell, right? Masking the boundary, and then just go around the cell periphery. So we're only looking at the cell periphery. We're removing signals coming from the cytoplasm. And then you just can mark out different regions of the cell, right? You can clearly see the leading edge, the lateral region, and the trailing edge is right about here. And from here, you can extract, for example, the ratio of the the membrane voltage coming from the ratio at the cell front, the cell back at the lateral region, right? So the difference is quite stark. And then we can also do some validation. We've done some more validation in the paper. So here, for example, you can just take the average of around the cell periphery. I take an average around the cell periphery and then get the memory voltage for a single cell. And then you can also add some ionophores like valinomycin. Valinomycin allows the potassium to go through the membrane. This makes the cytoplasm more negatively charged, so hyperpolarizes the cell. And we see that in this with the JEDI probe. And then if you add gramicidin, which punches a hole in the cell membrane, allowing all kinds of ions to go through, this depolarizes the cell, right? And we see these results with this ratiometric probe. It's interesting that we do see a slight voltage difference between front and back of the cell, as predicted by the osmotic engine, right? So the leading edge and the trailing edge, there is a slight voltage difference, maybe not very statistically significant, but this really is nothing compared to the difference between the leading trailing edge versus the lateral region. Is much more prominent. So osmotic engine does not explain the gradient that we see. And we were puzzled by this. We were wondering, is there some new physics here, right? It's pretty interesting because at least the standard biophysical model does not predict the presence of a gradient, but we do see it very, very clearly. Okay, so to solve this puzzle, we kind of dive into the idea to the question of what is the electrical environment of the cell membrane, right? So here's a lipid bilayer. You've got, different kinds of lipids, including phospholipids, which are acidic, so they're typically negatively charged in the lipid environment. So negative charged lipid would then be neutralized by the positive counterions, right? Potassium, sodium in the cell membrane, in the cytoplasm, right? So this forms a diffused bilayer. Why is it diffused? Well, this just comes from thermal energy, right? So because of thermal fluctuations, some of the ions will be detached. So these ionic lipids, the charges are not 100% neutralized simply because of thermal fluctuations, some small detachment of these positive ions from the negatively charged groups. So then if you then plot the membrane voltage as a function of space across the lipid bilayer, you expect this kind of profile. So at infinity, outside the cell, membrane voltage is hopefully zero, right? And then as you approach the cell, because of the presence of charges, it will deviate away from zero. And then it declines across the membrane, and then it reaches the inner leaflet where a lot of negative charges are present. This actually further declines further. And then it goes back to the cytoplasmic value in the far field in the cytoplasm, right? So this is the expected membrane potential profile across the membrane. And then if you ask, what is the membrane potential? It actually becomes one actually has to ask a more precise question, because if you mean the far field membrane potential difference, this will be the cytoplasmic potential, right? And by the way, the cytoplasmic potential relative to the outside, this is what's measured by the patch clamp experiment, because patch clamp directly accesses the cytoplasm and compares that with the far field membrane potential. Whereas if you look at a different region across, in this graph, this membrane potential phi local is local across the membrane, right? This phi local can be different from phi cyto because of the presence of these negative charge groups. I want to emphasize that membrane proteins, including JEDI, really only feels phi local. In other words, the membrane potential across the lipid bilayer. It doesn't really feel the cytoplasmic potential, right? So Jedi reports on this potential, and also this is what is also regulating all the other membrane proteins, presumably because they all sit in membrane, therefore feeling the local potential. So I like to say that phi local is more important because this is actually what's regulating what proteins are doing in the membrane. So then if you look at the lipid composition in the cell membrane, there are a lot of sort of neutral lipids, right? Cholesterol and so forth, glycolipids. Phospholipids are negatively charged. Sorry, some of the phospholipids in there. Some of them are neutral, PC and PE, they're neutral, but there are also negatively charged phospholipids. Out of these phospholipids, there are two which are dominant, PI, phosphatidylinositol, right? PS, which is phosphatidylserine, these are the most abundant. And then PI.
Okay, I found this on the web for these are the most abundant. Check it out.
Siri was interested in this talk. Let me shut it off. PI also is a very important lipid because it can be phosphorylated into PIP, PIP2, and PIP3. And some of you, if you follow the cell migration field, you probably have heard that PIP2, PIP3 are involved in cell polarity and migration. So these are highly negatively charged phospholipids. PIP3 has a -6 charge. PS has -1. The charge is -1, so it's not very highly charged, but it's very abundant, right? These two other lipids are more charged, but they're less abundant. So we decided to take a look at how these lipids are distributed in the cell using, again, ratiometric probes, right? So here's the PS indicator. So in order to get a ratiometric reading, we express CAX GFP in the membrane, and then you express a fluorescent protein that will bind to PS, or PIP3 in this case. So for PS, this is LAC-C2 M-cherry. And then for PIP3, it's AKTPH M-cherry. And then you simply take both channels and take a ratio. And then very quickly, you see that the degree of anisotropy we see in the anionic lipids, right, exactly mirrors the anisotropy we see in Jedi. It's uncanny, right? The difference between maximum and minimum in the ratio is about 0.85, and we see exactly the same thing for PS and PIP3. So the hyperpolarized lateral region of the cell seems to correlate with higher concentration of anionic lipids in the lateral region of the cell. They somehow accumulate there. And then the depolarized region, the front, is because we have less anionic lipids in that region. Curiously, there is no front and back difference between the leading and trailing. Right? Remember, there's a slight difference between leading and trailing edges of the cell in the Jedi signal, but there is no difference in the front and the back for PIP3 and PS. So maybe that the front and back difference is due to the fact that there's net ionic flux out of the cell. Okay, so is there any new physics? Now that we see that there is a heterogeneous distribution of ionic lipids, can it be explained by known physics, right? So the known physical model will be the Poisson-Nernst-Planck model, which just describes the potential. This is Gauss's law, right? The potential coming from all the charges. So we have charges, lipid charges, mobile ion charges. So this is Ci is the concentration of mobile ions, and then Z are their charges. And then Qn, these are impermeable charges like proteins, right? So that's in the cytoplasm. And this equation must be coupled with ionic flux equations, right? So ions can diffuse. So you have a diffusion of them driven by potential gradients. And also there's additional flux maybe across the membrane for these ionic fluxes. So these two equations then constitute the Poissonneur's Planck model with the modification of negatively charged lipids. And then if you just set up a model, here's the membrane. There is some permittivity differences in the membrane versus the outside, right? So dielectric constant is lower in the membrane. So this is the permittivity profile across the membrane. Here's a lipid charge profile. We put this in by hand, right? So you have a slightly higher negative charge in the inner leaflet. Here's the sodium potassium concentration outside inside. There's a gradient. And also we can put in by hand various different types of ionic fluxes at the boundary. And then you can also put in by hand the concentration of impermeable molecules, other charges. So if you then solve this set of equations, you'll find indeed, if you have excess anionic lipid in the inner leaflet, this will influence the local membrane potential. You see that as I increase the concentration of anionic lipid in the inner leaflet, the membrane potential gets more and more negative, and then that changes the local membrane potential across lipid bilayer. On the other hand, so this is a plot of now the membrane potential in the x direction, right? So this plot here is across the y axis, which is perpendicular to the membrane. So if you do the same plot across the x-axis, so we put in by hand the negative anionic lipid distribution, which is just this sine function, right? And then you can see that the resulting membrane potential follows the anionic lipid distribution in X. And then you can also, put in any kind of polarized distribution of ion fluxes. This does not generate any spatial patterns, as we predicted earlier, because of rapid diffusion of ionic charges. So this leads us to conclude, at least the model is consistent with the idea that ionic lipid is responsible for the voltage distribution that we see across the cell periphery. Okay, so to test this idea, the fact that we think that ionic lipid is what's generating the voltage gradient, we then use, and the lipids are trafficked, right? That they're being consistently turned over, so lipid trafficking processes occurring in the cell. So we decided to test this out using FRAP. So this is just cell expressing the PS indicator, and then we just FRAP or fluorescently bleach different region of the cell, leading versus the lateral region. And then we can look at the FRAP recovery, right? Because if you bleach the fluorophore in that region, the new fluorescence signal you see there was coming from presumably lipids that's either diffusing into that region or being delivered via vesicle trafficking into that region. And then if you just look at the FRAP recovery curve for the leading versus the lateral, we see a dramatic difference. The lateral region recovers fairly quickly and completely. Some of this is coming from diffusion, but you see the leading region of the cell, the recovery is incomplete. And then additional evidence is that if you block lipid trafficking using dinosaur is a dynamin inhibitor. That dynamin is involved in generating vesicles, generating curved membrane. If you block it, you can completely remove the differences in recovery curve, right? So this difference in the FRAP recovery is due to lipid trafficking, at least from this experiment. And then not only dynamin blocks recovery of PS, it removes the gradient in all three things we looked at, ratiometric probes we looked at, right? So JEDI, PIP2, and PIP3, and PS, all of them, this is a plot of uniformity, which is the difference, the maximum difference divided by the average, and you see that all of them becomes more uniform if we use dinosaur, further supporting the idea that basically they're the same thing. or the voltage grade, the membrane voltage is basically similar to anionic lipids. And then you can also, do electrostasis experiments. You can apply voltage for cell expressing JEDI and PS, ratiometric probes. In this case, right, you see that, so this is JEDI. Initially, you see that JEDI is, the protrusive region here is red. It's more depolarized. The protrusion is in this direction, right? You turn on electric field, and you see over time, JEDI becomes more depolarized, and the new protrusion towards the negative anode in the electric field. And then you can do the same experiment with PS. So this is PS here. You see that PS is more abundant in the protrusive region here, right? That's blue. Sorry, more abundant in the red region, the lateral region. That's red. Red is higher. And then if you turn on electric field, it will reorient, become more abundant in this new lateral region. That's away from the protrusion. Okay, so at least this set of experiments show that lipid trafficking, which sets up, we'll put different lipids onto the membrane, also take in lipids in different parts of the membrane, is what's setting up this gradient that we see in the anionic lipids, right? So presumably there's more lipid being taken in at the leading edge, at the protrusive region, right? So that's taking away anionic lipids. And then there's more anionic lipids that's being deposited or put onto the lateral region of the cell. This then generates higher concentration. Of course, there's diffusion of lipids, but the steady flux of in and out in different regions is what's setting up the gradient. And I didn't show this, but myosin 5, right? This lipid, lipid trafficking requires things like motors, which deliver the vesicles to the membrane. If you block that, also you can remove some of the gradient that you see in both JEDI and also PS and PIP3. Okay, what about ion channels? So we see that the gradient is correlating with ionic lipid distribution. Ion channel, heterogeneous distribution of ion channels should not generate any gradient, right? At least not from the action of passaging ions into the cytoplasm. Do they do anything to the JEDI signal we see? And in particular, we're kind of interested in this ion exchanger called NHE, the sodium hydrogen exchanger. This one, on the surface, it seems to should not influence the membrane potential because it exchanges one sodium with one hydrogen. It doesn't change the charge state of the cell, right? And we know from other studies that it is concentrated at the leading edge of the cell when the cell's migrating in channels. So does this ion exchanger influence membrane potential? It turns out it does, okay, actually quite dramatically. So, and it's because even though it doesn't change the charge state of the cell, it does influence potassium exchange, right? It changes sodium concentration, which influences sodium-potassium exchanger, and that ultimately influences potassium concentration. So if you use EIPA to inhibit NHE, you will lead to cell hyperpolarization. And but if you increase potassium permeability using valentomycin, this effect goes away, right? So we do see that NHE is influencing membrane potential, at at the global scale by influencing potassium flux. And that can also be explained by a computational model. Okay, but what's surprising maybe is that not only changes the overall membrane potential, it actually changes the membrane potential in different regions of the cell differently, right? It hyperpolarizes the cell, but you see that with EIPA, if you add EIPA, it quickly elevates the leading edge much more than the lateral region. It hyperpolarizes the leading edge more than the lateral region. If you leave it in longer, there's some adaptation processes happening, right? But it does also influence the membrane potential anisotropy that we observe. And maybe this is not too surprising because not only NHE passage ions across the membrane, it also is associated with actin dynamics, right? It is physically connected with actin cytoskeleton, and it's also involved in vesicle formation. NHE is involved in generating vesicles. And there's been a lot of work that shows it interacts with things like PI3K. So it is also involved in lipid trafficking and also cell polarity. So in that sense, not only influences potassium flux, it has another role of influencing cell polarization. So it behaves almost exactly like latrunculin, which disrupts actin cytoskeleton, right? So everyone knows latrunculin is involved in cell polarity. Actin cytoskeleton is involved in cell polarity. And if you look at JEDI, PIP2, and PIP3 and PS, all of them, if you add either latrunculin or EIPA, you can remove the anisotropy that you observe. So they are part of the cell polarity mechanism. Other ion channels also predictably change the polarization state, the membrane potential of the cell, right? So this is the average membrane potential around the cell. So these are different ion channel inhibitors. You see some will hyperpolarize the cell, right? Bumetanide, DIDS inhibits anionic exchanger. And then some ion channel inhibitors will depolarize the cell, like oibane, which sets up the potassium gradient. So if you inhibit it, cells become more depolarized. This all makes sense. But we also see that these inhibitors also influences polarity, right? If you inhibit these ion channels, you will change the uniformity across the cell membrane. So that says that ion channels, together with NHE, are all potentially part of the cell polarity mechanism. Cytoskeleton, F-actin, is involved in trafficking lipids, of course, but also membrane proteins in the lipids, right, including ion channels. And the activity of ion channels somehow also influence this trafficking process. So there's a tightly coupled network together with cytoskeleton ion channels, which influence cell polarity and membrane potential distribution. And so not surprisingly, some of the mechanobiology phenomena also involves membrane potential, right? Like, for example, you see that if you put cells on different substrates of different stiffness, we know that cytoskeleton organization is changed when you change substrate stiffness. This also leads to a membrane depolarization on a softer substrate, right? And then just the physical act of mechanical stretch. So if you just put the cell on a flexible membrane, you stretch it, you'll see that cells actually hyperpolarize. So 3T3 will hyperpolarize after a few minutes, and then recover. H1080 also has has almost a null response, doesn't seem to respond to mechanical stretch. So cell mechanics or mechanobiology questions is also influencing membrane potential. So this is self-spreading. You just take the rounded cell and you put it onto a substrate. It starts to spread. As it spreads out, you see that it depolarizes. It's very, very obvious. And this depolarization probably has to do with cytoskeleton activating NHE during the spreading process. Mitotic swelling. Here's another thing which is quite interesting during cell mitosis, right? The cell rounds up. We know that mitotic swelling, the cell volume actually increases. And then we see a corresponding hyperpolarization that happens during the mitotic swelling. And this potentially points to a mechanism of mitotic, or the reason for mitotic swelling, right? Probably is corresponding to a potassium concentration increase, which allows chromosome to reorganize. Remember, potassium and other counterions are involved in DNA organization. So if I want to reorganize my chromosomes to separate them, I need to change my cytoplasmic ionic concentration, potentially, through this process. So if you just look at the cell in general, you see a negative correlation between cell area and membrane potential. So if the cell area is large, the cell is generally more depolarized. Sorry, hyperpolarized. Larger area is larger cells typically is more depolarized, and then smaller cell area corresponds to a hyperpolarized state. So there's some coupling going on between cell shape and also membrane potential. And then, in the closing few minutes, I'll just tell you a little bit about higher level organization in things like epithelia. In epithelia, it's not just a single cell that shows membrane potential heterogeneity, but also there are domains in epithelium which shows higher order structures, right? So this is, for example, a confluent MDCK epithelium. First, you see that there's also a cell area versus membrane potential anti-correlation, right? Smaller cell area correspond to more hyperpolarization. This is just from a single plane in the epithelium. But then if you look at the membrane potential cell area correlation function, right? So this is now the correlation between a membrane potential and the cell area plotted as this pairwise distance between the cells. I told you they're typically within the same cell is negatively correlated, right? So within the same cells is negative correlation around 10 nanometers, 10 microns or so, right? But then as you go further out in distance, this is now other cells, other neighboring cells, it becomes positively correlated now. So there's this long range correlation between cell area and membrane potential, and also a long range correlation just in the membrane potential itself, right? So this is membrane potential of one cell, how it's correlated with another as a function of their distance. And you see that there is a positive correlation up to some point of 100 microns. So that's several cells in the membrane, in the epithelium. And this correlation actually persists and also changes over time. So this is a plot of this correlation function as a function of distance for different times. And you see that as time changes, the correlated domains also change. This, in fact, undergoes oscillation. This domain kind of gets bigger, smaller, with a time period on the order of 40 minutes or so. We're still investigating this question. We know that these domains are set up by things like cytoskeletal activity. If you add white compound to this, you remove this domain and these correlated domains, and also you remove this oscillation. But clearly, in epithelia, there's higher order correlations or higher order structures in the membrane. in the membrane potential. And so that was in plane of the epithelium. If you look at just from the apical basal surface, there's also a membrane potential difference, right? So this is just looking at the cell, but now in a Z-stack. And if you quantify the membrane potential at the basal surface versus the apical surface, there seems to be a difference. The basal surface is more depolarized. And then we also know that this depolarized basal surface is correlated with enriched PS localization in the apical surface, right? So it's more hyperpolarized in the apical surface because there's more PS there. And then finally, maybe the first step in morphogenesis, this is epithelial domes, right? Epithelial domes are water-filled domes that comes about when the epithelium matures. So water is being transported into the dome, generating this bump, right? So you see that this empty space is in fact a bump. And in epithelial domes, water transport seems to also correspond to a change in the apical basal polarity of membrane voltage. So in the dome, for cells in the dome, there's less apical basal membrane voltage difference than from cells from outside the dome. So that says that there's a polarity difference happening for cells that are in the dome versus the cells away from the dome. So I think I've reached my time limit. I really should stop here. So here are some of the tentative conclusions we have, and I'm happy to answer any questions you have.
So usually Yuping would now pick up the questions. Sorry, I'm like for the silence, but maybe he has a sound or connection issues. So then I will jump in him for him right now spontaneously. So thanks a lot for a wonderful talk. We usually put the questions in the chat, and Tomoko already has the first question.
Oh yeah, and then also
Yeah.
Miss Lana is raising hand. So
All right, then let's give the first question of the semester to Mirana.
Yeah, sounds great.
Hi, can you hear me?
Hi. Yes.
Thank you for your very interesting talk. I really enjoyed it. I have several questions, but maybe the one that is more relevant to the final part of your talk is about the motion of cells. So you showed that in the lateral side and the protrusion side, there's a a large difference between the membrane potential. Is it known that if this membrane potential plays a role in the cell movement, or is it a consequence of actin or cytoskeleton, higher cytoskeleton or higher actin concentration at the protrusion side?
Yeah. I think the answer is that yes, the membrane potential definitely plays a role in cell movement, cell migration, simply because it influences, ion channel activity, which we know is also part of the cell movement apparatus. It's not easy to generate a cause and effect description of what membrane potential does because there's a feedback going on, right? Membrane potential influences ion channel activity, but ion channel activity ultimately also influences membrane potential. So it's kind of like a feedback homeostatic situation. But yes, I think membrane potential definitely influences cell migration. And also a lot of experiments have shown that if you apply external voltage or an external potential, that can change membrane potential that can change cell migration, cell polarity, right? We saw that also with JEDI and anionic lipids. So they do respond to external electric fields, which presumably influences anionic lipid distribution, but also influences ion channel activity.
But it's like it's not the main drive of the cell motion, right? Like if we somehow manage to make this homogeneous across the membrane everywhere in the cell, how much of the motion are we getting rid of? Like how many percent of motion comes from that?
Yeah, so that's a hard question to disentangle because to make it homogeneous, you have to inhibit things like NHE and actin cytoskeleton. So to make it homogeneous, you have to remove cell polarity. But if you remove cell polarity, the cell's not going to move. So I guess it's all part of the same system, right? The way I think about it, it's a network where everything affects everything else. And it may
Yeah,
be incorrect to use a
no,
cause
but
and effect mechanism.
Yeah, I was thinking maybe with the anodes, with the inhomogeneous electric field or something, if someone manages to make it homogeneous and look at, but it's not, yeah, it's more complicated. Okay, thank you. And for the ion channels, is it that the activity of the ion channels is different, or is it the distribution also gets changes across the different regions in the membrane?
There is heterogeneous distribution, but at least from our understanding, heterogeneous distribution should not affect the membrane potential distribution because ions diffuse rapidly. But we do see that because ion channels interact with lipid trafficking, it is also influencing the lipid distribution, not because of their fluxes, but because they interact with the lipid trafficking process.
Thank you.
Yeah.
And then we have a question from Tomoko.
Yeah, thank you so much, Sean, for a great talk. I was wondering if this probe gets influenced by the curvature of the membrane, or if it can be completely separated.
Yeah, so yeah, that's a good question. And we don't have a definitive answer because it's actually hard to quantify membrane curvature, I guess. You probably know membrane is, there are a lot of wrinkles in the membrane naturally. So and we don't see that in the microscopy. So we don't exactly know how membrane curvature is potentially influencing Jedi signal.
I see. For example,
So
could
yeah,
you maybe purify this probe and then form the in vitro vesicle of different size, for example? And then the reason why I was thinking of if you could get absolute value of the local membrane potential in such experiment, but if the curvature affects the probe signal, it's probably difficult.
yeah,
So
yeah. Yeah, that experiment is possible. Actually, I think Jen Shi is in the audience. You can probably make a vesicle express with Jedi in there, and then just look at how different vesicle radii correspond to. That's something that his lab can do. We can't really do that, but I would love for him to check it out.
Okay, that's very good. Thank you so much.
Yeah.
Okay. Then Yuping is apparently complaining about his new computer, which is not better than his old one, apparently. So I was really struck by that one image you showed of your cell that clearly shows like these two distinct domains. And I was just, wondering, it's not like a gradient, right? It seems like either you're on or off. And so I was just wondering how you imagine how this can happen, that you can compartmentalize the membrane like this. Yeah.
The way I think about it is not really compartmentalizing. I think about think about it as like a transport process where I'm shoving in stuff on one side, right? And I'm taking away stuff on the other.
Yeah.
So that continuous transport process naturally sets up a gradient.
So exactly, but So that's what you were saying here, right? But then when I look at this image, it looks more like there's nothing in the front, and then there's something like where the nucleus is afterwards. There is something, right? So it's not like a gradient, but kind of a switch almost. Do you see that
Yeah,
often,
I see.
or?
Yeah, I see. Yeah, I mean, we do see these, distinct domains. There may be higher order complexities that we're not probing here. Maybe there are lipid domains in there that prevents diffusion of certain types of lipids also. Yeah, that's certainly possible. Yeah.
Yeah, I don't know, or like I don't know. You were mentioning also the vesicle trafficking, and yeah. This is very very curious.
Yeah.
Thanks.
Yeah, the vesicle trafficking, we also see anisotropy, right?
Yeah.
But maybe there are finer details that it's being targeted to a certain region due to certain
Yeah.
properties.
Yeah.
Yeah.
Yes. Sorry, I want to jump in to add a little bit comments to Sean's answer because another possibility, because you have seen that when the epithelial forming this water dome, that means there's an apical to basal transport. There's a huge change in this plurality.
Mm.
Therefore, what Sean kind of describing may not necessarily occurs across the epithelial axis. It might be apical to basal. You might have a little bit of pumping at the one region that's more than the
Mm.
other. Therefore, I can generate this domain.
Thanks. Then, Yuping, you still want to ask your question? You said you were going to decide after I asked mine. And because he probably has to write his answer, in the meantime, we will take Rose's question.
Hey, Sean, great talk.
Hi.
Question for you about lipid phosphatases. I'm wondering if you've looked at the effect of lipid phosphatases on these gradients, and what would you expect to find if you haven't?
Yeah, that's something we're looking into right now. So PIP can be converted. So we do see that if you inhibit PI3K, which you probably know that PI3K converts PIP2 to PIP3, right? We do see that it influences memory potential. It actually makes the cell, I can't remember exactly, more hyperpolarized. No, it has the same effect as actin inhibition. Anyway, it does influence memory potential significantly. And the effect is difficult to entangle because it's not only changing the charge state of the inner leaflet, but also is influencing cell polarity. So it's also influencing lipid trafficking.
And I guess presumably cell migration too, right? I mean,
yes, PI3K is known to organize cell migration. Yeah,
so
chemotaxis.
blocking its activity would
Right.
have the opposite
Right.
effect. Okay.
Right.
Thank you.
Yeah.
Okay. So while we're still waiting for Yuping's question, Jinrong has a question.
He says he can't speak, so I will read his question.
Yeah.
I'm
See, I didn't read to the bottom.
Yeah. He says, I'm curious what's the biological function of higher anionic lipid at the lateral side, or is it just a consequence of higher actin filaments with less specific function? I don't have a good answer. I'm not sure. I'm not sure what the reason would be. We see it both in 2D glass and 3D collagen gels, right? So it may just be a consequence of polarized actin cytoskeleton and its activity of and its mechanism of directing vesicle to different places in the cell. So, yeah.
And Yuping wrote his question, could it be that instead of differential transportation, anionic phospholipids, I moved, are pinned down by proteins that have preference for high curvature? Are the lateral regions being more stretched?
Yeah, that's also a good question. Yeah, like I don't know. It's certainly possible, right? Although I'm not sure the lateral regions are more stretched. It appears to be more taut in in microscopy, but if you, I mean, there's been a lot of work that showed that membrane in cells are very slack, right? There's a lot of slack in the membrane. If you zoom in at a lower length scale, you'll see lots of, lipid undulations. So, and we know membrane tension is generally pretty low. If you do have tension gradients, it should equilibrate very quickly because tension gradient will drive lipid flow. So while some proteins may be localized in the membrane, which then leads to localized ionic phospholipids, that's certainly possible. I think the question of membrane curvatures localizing proteins is more complicated because there's just a lot of curvature in the membrane at the scale of proteins, at the scale of a few nanometers.
All right. Thanks a lot. So if there are no more questions, let's give Sean a big hand for a great talk. Thanks a lot for being here.
Thank you so much.
And
Thank you for giving us the opportunity. And I'm happy to answer any other questions offline if you just want to shoot me an email.
Wonderful. Thanks a lot. And see you all back in 2 weeks.
All right.
Bye-bye.